Good morning, all my teachers, dignitaries, learned relatives, all my friends. I am Sridevi Patil from second year BFC. I feel privileged to welcome you all on this auspicious occasion on behalf of our zoology department. Before we start the gathering, I would like to give a brief introduction about our KLE Society. The KLE Society Belagavi started since 1965, group of 7 and dedicated graduates, respectfully called Saptarishis. The KLE Society disseminates the knowledge in all spheres of education, KG to PG, for overall personality development to the students. Under the leadership of visionary chairman Dr. Prabhakar P. Koresar, ex-MP, the number of institutions has elevated up to 298 in various fields of education, including research in India and abroad. Coming to our KLES Nizingappa College. KLE Society S. Nizingappa College, established in the year 1963, it is one of the premier institutions under KLE permanently affiliated to the Bangalore City University. The college takes pride in its distinguished alumni in all walks of life. Coming to about our zoology department. The Department of Zoology came into existence in the year 1967, currently affiliated to Bangalore City University, offering BSc with CBZ, CZBT, CZ and BZ and ZBT combinations. Department has state-of-art museum, well-equipped laboratories, central research laboratory. The department designs and executes several student-centric activities, national and international seminars, workshops, lecture series, faculty department, programs and conferences. Faculty of the department facilitates innovative student research projects, and I'm proud to be a KLE student. Now I would like to tell two words about our today's national conference. The Evolution of Infectious Diseases Conference supports research on the evolutionary and organism and social drivers that influence the transmission dynamics of infectious diseases. The theme of submitted conference is quantitative, qualitative, mathematical, or computational understanding. The pathogen transmission dynamics. This conference focuses on understanding the molecular structure and function of known viral, bacterial, fungal, and parasitic pathogens. Now let's begin our program with the blessings of God. I would like to welcome Shalini from Zoology Department to sing a song.
ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ
Shalini for giving a special song. ಹಚ್ಚೆಯು ಕನ್ನಡದ ದೀಪ ಕರುನಾಡ ದೀಪ ಸಿರಿನುಡಿಯ ದೀಪ ಬೆಳಗಲ್ಲಿ ತೋರುವ ದೀಪ ಎನ್ನುತ್ತಾ ಐ ವುಡ್ ಲೈಕ್ ಟು ಕಾಲ್ ಅವರ್ ಗೆಸ್ಟ್ ಟು ಲೈಟನ್ ದ ಲ್ಯಾಂಪ್. Thanks to all my teachers and sirs. Now I would like to welcome Tejaswini V. Nandri ma'am to welcome our guests.
Good morning, all. Good morning. Good morning, everyone. Present here. It's my privilege to welcome you all for today's national conference on evolutionary biology and infectious diseases, organized by Department of Science. We are very lucky to have very talented and competent personalities amongst us today. Dr. Devraju K.S. and Professor and Chairman Department of Biochemistry, Karnataka University, Dharwad, and Dr. Shabir Ahmad, Assistant Professor in Biochemistry, Basic Sciences and Humanities, University of Jammu and Kashmir. So they are amongst us today as the resource persons. On behalf of KLE Parivar and on my own behalf, a warm welcome to esteemed resource persons, Devraju sir and Shabir Ahmad sir. Thank you for accepting our invitation. Welcome. I request our principal to welcome Dr. Devraju sir by offering a token of it. Dr. Shabir Ahmad sir is online speaker of second technical session. I welcome you, sir. It's my great pleasure and honor for me to welcome our beloved principal, Dr. Arun Kumar V. Sanathan, sir, on behalf of the entire administration, faculty, and students. I wholeheartedly welcome you, sir. Sincerely thank you for accepting our invitation in spite of your busy schedule. I am glad to welcome Professor Chandrasekhar Appa, HOD of Mathematics and IQAC Coordinator. And I welcome our Principal of Pharmacy College. I wholeheartedly welcome to all oral and poster presenters and the participants from different states like Himachal Pradesh, Uttar Pradesh, Telangana, Kerala, West Bengal, Tamil Nadu, Puducherry, Maharashtra, and from different places of Karnataka state and also from Delhi for being a part of this national conference. I take this opportunity to welcome all the HODs, staff, and students of this college for this conference. I ensure all of you acquire a fruitful knowledge by the end of this day and make use of this conference to the best of your abilities. I wish you a most fruitful day with interesting and stimulating discussions. I request all of you for your kind information, kind cooperation throughout the day. I congratulate all the oral and poster presenters. Wish you best of luck.
Now I would like to call Swasini ma'am to introduce our guest.
Sorry for the interruption. Good morning to one and all here. To audience, I would like to make it clear that this is the national conference, and actually we are organizing in a hybrid mode, offline as well as online. So on behalf of the entire the KLE SNC Parivar, I would like to welcome all the resource persons and the participants and even the candidates who are going to be present, their oral presentation of their research findings. So I once again welcome to all the offline and as well as online participants of this national conference. Thank you.
A very good morning, one and all present here. I am privileged and humbled to take this opportunity to introduce our today's chief guest, Dr. Devaraj Kote. Sir was born in a small village, Kuramkote, that is the Kotegeri Taluk, and he brought up in Tumakuru. And Sir completed his bachelor degree from SSFGC Tumakuru in 1996. Later moved to Bangalore for higher education and obtained master degree in biochemistry from Bangalore University in 1998. He qualified JAVET NET conducted by CSIR in 2000 and also qualified NET conducted by Agricultural Scientific Equipment Board in 2001. Sir completed his doctoral studies. Sir completed his doctoral studies in Department of Neurochemistry, National Institution of Mental Health and Neuroscience, Bengaluru, entitled Studies on Protein Phosphatase, Substrate Specificity and Assay Method. Sir completed several certificate courses like core breeding and management of laboratory animals from humans and bioprocesses engineering and NMR spectroscopy, principles and applications to biochemical systems from Indian Institution of Science, Bengaluru. Sir has more than 20 years of teaching and research experience. He completed 5 different major research projects funded by UGC, DST, BURIF and RGUHS. Sir has successfully guided 5 students for their PhD program and currently 4 students are working under his guidance. Sir has delivered more than 25 invited lectures in our various universities, colleges, and schools, organized more than 20 workshops. He written 9 book chapters for MSc and published 40 national and international peer-reviewed journals and presented around 56 national and international conferences of his work and visited Florida, USA in 2010 and Singapore in 2016. He has obtained short-time research training in the University of Chicago, USA in 2010 and recently the University of Baltimore, USA in 2022. He awarded Raman Postdoctoral Fellowship in 2013 and Adhyapak Bhushan Award from Shikshak Sangh and Best Paper Award in the National Conference organized by the Karnataka State Higher Education Council and also from Tamil Nadu University. He has delivered 14 video lectures in Mysore University under UGC program. He also delivered 8 lectures UGC Academic Staff College of various universities. He is also life member of several research institutions or societies like Neuroscience, Washington, USA, and Society of Biological Chemists and Bengaluru, and is a committee member of ICMR. He has a collaboration research work with the Ayurveda College and Medical College, NIMHANS, and CFTRI, and DRM, HANS, and many more institutions. And currently, he is a professor in the Department of Biochemistry and also the director of the Skill Development Center, Karnataka University, Dharwad. With this brief introduction, I welcome Dr. Devaraju Kote. Welcome you, sir. Once again, welcome you. Now I call Dr. Kote sir to present.
Once again, good morning, everyone. At the outset, I'm deeply happy and privileged to be associated with the All-Day National Conference. Let me express my deep sense of gratitude to the greatest visionary of the education, is the Professor Professor is the chief patron of this society, educational society, and also Mahantesh Kodhiman, ex-MLA sir, is the patron of this education society, and Sri Shankarana Munavalli sir, he is the director. So I think with the great visionary such people, I think the great education society could come up and cater education to the large population of the society. So the moment when I got a call from my friend, Dr. Naran Swami sir, so I know him when I was working with the Dhananand Sagar College. So he said, we are organizing a national conference on evolutionary biology and infectious disease. I think this is really out of box for me to talk. But anyways, the invitation is with me now, with a show of hands. Right? Again, for this organizing such a wonderful one-day national symposium, the efforts cannot be forgotten. So I greatly acknowledge with Dr. Arun Kumar Panampanavar, principal, who really giving all encouragement for conducting such a wonderful program. At the same time, I really congratulate the Department of Biotechnology for Fluoroscopy and the head of the department, Tejaswini Nandini, and organizing committee members, Subhasini Pudapuri, and again organizing committee members, Narayan Swami sir. I cannot forget that because the invitation is through him. So, as already I mentioned about, it's not a cup of my tea, but still, on the evolutionary biology and infectious disease. We need to combine both an evolution and as well as infectious disease. This is the biggest task given to me. I don't know how much justice I will do it, but nevertheless, during the COVID days, I had a lot of time as everybody had. So I thought of preparing some awareness program for social, there was a larger society, and as well as particularly the student fraternity. Those clients which are pertaining to only COVID, which I made, and I delivered even an online talk also, and I shared in the YouTube. You can also watch it and see the same talk. But what the difference which I need to make here for the evolutionary biology and as well as the infectious disease, some thoughts went into my mind, and I have prepared some of the slides with my experience to share those things with you. Hopefully, you will enjoy this 40 minutes of slide along with me. Corona, Corona, Corona. It has bothered us so much. Right? This was the title which I made earlier, Corona, but I made it now. COVID-19 was a molecular evolutionary challenge to the world. Just to bring evolutionary into a chair. Nothing more than that. So the moment evolution, when we hear about, so all of us know, maybe a high school days to a college days, we would really understand about Darwinian theory of evolution, right? So how a man has been made from the monkey. Now, from where to understand this evolution, so probably you're all expert, by reading the Darwinian theory and saying that ape like ancestors that turned out to be a man now just standing before you. Right? So the evolution has not taken place only with that level, just as just now when we were discussing with principals were telling me that this is a heterogeneous population. And initially, Narayan sir also said about only the BSc students are there along with the teachers. So the evolution is not happened all of a sudden. Let's say it was a theory being put forward like how the chemicals are being evolved. So from there, with a couple of slides, I will take it further. Somewhere in 1924, O'Hare officially put forward as influential a theory that life on Earth developed to a gradual chemical evolution of a carbon-based molecule, which is called as a primordial soup. At just about the same time, the British biologists also put forward the same similar kind of a theory that they both independently proposed this theory, primordial soup. So these are like very important for the chemical evolution as such. But however, these are the theories that while as we make an hypothesis, similarly the theories have been built with a logical expression, but experimental proof is really required. So here is the biochemist called Stanley Miller. So Stanley Miller, he did some kind of an experiment along with the urine. So you can see that the urine is a senior most professor and Stanley was a much younger to him. So again, probably together, Guru and Shishya, they were together doing the work. And they have come up with exactly the model, a chemical experiment, which simply mimic with how exactly the prebiotic synthesis do happen. So there was some set of gases were present. So those gases were kept over here in the cylinder on that side. You can see over there. So which was kept with under electrodes. The electric spark was given to these gases, and now the molecules have been synthesized. So this is what exactly the chemical evolution. So practically proved, the theory of chemical evolution was practically proved to say that there is an evolution which is happening from molecule to, let's say, gases to a molecule, right, with the appropriate condition as such. Right, as we all know, like in a POC, you might have heard about, when there is a rain comes, nitric oxide combined with the water and then becomes HNO3. It is a seriously disaster which happens to many of the monuments, right? So these are all we have understood. Only similar kind of an experiment being kept, and later these small molecules are joined together to form a complex molecules, lipids and polypeptides, and then polynucleotides and nucleotides. All of you know that nucleotides are the one genetic materials which you talk about. They are the ones which really do things, right? So now the question is this. So as primitive, the gases becoming the compounds and then complex molecules. Complex molecules, as we all know, the biomolecules are nucleic acids, RNA, right, DNA, and the proteins. These are the complex molecules, and these molecules are there already now. They are here with us in all part of life. Let's say. So now the DNA comes first, RNA comes first, or protein comes first. This was a big buzz at the time. Okay, the people started thinking of what would have come first, evolved first. So there is a theory which is being put forward by Gilbert somewhere in 1986. Worldwide people have accepted there was an RNA word. So as we have understood about the RNA could bring about some kind of genetic information, a flow of genetic information, right? So as we know now the flow of information, how does it happen? Is that by the central dogma of molecular biology, right? So the information, the flow of information goes with the DNA to RNA, RNA to a protein, but it never happens on a reverse. That was the concept which was put forward somewhere in 1974 by Watson and Crick. No, Watson alone, not the Crick. But RNA was the one, or DNA was the one, because as we know now, the existing system now, the DNA is the first. That's the blueprint, which will transfer the information to an mRNA, and then the protein comes into picture, which are our work causes. But then how this RNA? So the concept of RNA, and sometimes people started arguing that, okay, without a protein, without an enzyme, how the DNA polymerization will happen? These are the kind of questions, right? And the DNA all alone, it cannot replicate. This is another difficult question. So, however, the catalytic property of an RNA being established very clearly. So now, the RNA could bring about, could be used as an information storage molecule, and as well as they work like a catalyst also. I mean that intermediate kind of what you call the property be shown exhibited by RNA. Let's say it can work like a catalyst, and it can work like information molecule also. So that's why the concept was accepted worldwide, and there was no RNA word was accepted too. But there are still a continuous argument to say that there is no RNA word. But we'll not worry about it. Now, when we say that the RNA, so if you look at the evolution, the unicellular organisms to a multicellular organisms. So this is the evolutionary tree, all of you might have been observed, right? Somewhere 3.8 to 4 billion years ago, so the unicellular organisms came into picture. And then slowly, these multicellular organisms, somewhere in 600 billion years ago, they have come over here. Let's say now the chemicals have been evolved from the gas, and then these chemicals started becoming complex molecules, and the complex molecules, they started arranging themselves based on their own properties. Right? You have sitting somewhere here, so your teachers are sitting here, and some of them sitting over there. Right? Let's say. So why did you sit there? If I ask a question, then it is a big question, right? So you will say that, okay, he is my friend and he is my batchmate, so on and so forth. In the same way, these biomolecules also, because of their molecular constitution, which is being present, they dictated to make a bilayer. For example, let's say the lipids, they have both hydrophobic in nature and hydrophilic in nature. They started turning the other way around in the aqueous medium. They started making a unicellular bilayer, let's say. So now there are proteins are getting inserted here and there, and some proteins have started settling in the cytosol. So now the cytosolic proteins and the integral proteins are well. So like that. So it all depends on the nature of the protein again, and then what it dictates. Of course, the genetic information has become well established when the DNA came into picture. The reason is that probably the RNA, if you consider, it is not quite stable in the nature. RNA undergoes easily hydrolysis when compared to the DNA. Maybe you can read some of the textbooks, particularly biochemistry, on why the DNA is quite stable than the RNA. Probably the other way around of answer is that because it is a double standard, 2 is always a best pair, right? 2 will be more stronger than an individual, right? Maybe that is also logically right. But chemically, it has been well proved that because of the ribosugar hydroxyl groups are there at the bottom, so they make the RNA unstable compared to the DNA. So that's why DNA has become what you call as the blueprint for life. So now the DNA started keeping the genetic information and started giving the slope information through an RNA. So that's how the RNA becomes what intermediate between the storage information and execution. So execution will be done by the proteins as such. But there are certain organisms when they started working, evolving, the molecules are left behind. So some DNA and some RNA, pieces of RNA, are left behind while they are getting evolved. So here we come with such molecules, we call it as a virus. So virus, as as a simple definition is that an infective agent that typically consists of a nucleic acid molecule in a protein coat is too small to be seen by light microscopy and able to multiply only within the cells of the host. So these molecules are dead when they are outside. When they get inside the host system, they become active. Really, such nature is the culprit, right? Always. The person who never does anything outside when he comes inside, he will do all unknown, right? And suddenly you will start beating them. That's what our kids do. Not necessarily, but when somebody is there outside, they will not. When they come inside, they will do all natural because we are parents or somebody. They will take an advantage. So now we have to tap them like that, because the virus are such a kind of behavior they exhibit. The outside they will not behave anything. They will be like a dead thing. And then once they enter into the system, they start utilizing our own machinery and they start getting replicated. That's what it happens. So there are varieties of virus molecules. So major criteria of a classifying human familiar virus. One of the articles we shared about pediatric infectious disease. This is the cartoon which I took from there. So the genomic genomic of the virus can be any of this, either DNA or RNA, either double stranded or single stranded or partially double stranded. See varieties, and we call them as a negative and positive sense and antisense, and they do have some kind of a capsid in a different fashion as such. Helical complex, icosahedral, and so on. So some do have envelopes and some do not. So all such different kinds of viruses are there. So here the statement given by E.O. Wilson is that the variety of the genes in a viruses exceeds that in all of the rest of life combined. If you take all varieties of life, multicellular to unicellular to multicellular, complete organisms if you take, if you really want to look at the varieties of the genes, they cannot match with the variety of the genes in the virus. That much of diversity in the virus genes have. So certainly they are very competent, very competent. Highly evolved multicellular organisms, let's say human beings, let's say. So humans are suffering by many of virus problems, right? Because the virus are having such kind of varieties. Maybe the simplicity and flexibility that I would tell you later. And little bit about corona, probably all this information you also would be knowing it. So just I leave with now the coronavirus, and which was too much of troublesome for us for a long time, right? Almost 2 years we got a degree easily, right? So many people do pray another corona should come, right? So that you will get easily degree, correct? Without reading. So this belongs to a family called Coronaviridae. Is a single stranded RNA virus. As I said, DNA, double strand, single strand, RNA, single strand or double strand, and even partially double stranded, all such things, varieties. That variety, this is the variety which is called as a Coronaviridae, which is the RNA virus, which is all about around 26 to 32 kilo base pairs in length, and the particles are typically decorated with 20 nanometer spikes. So the electron microgram of a spherical particle create an image called recent solar corona. Can you see here? This is not a virus as it is a solar corona. When the solar eclipse happens, there is a kind of what we call as a picture which we see where all around brightness and in the middle there is a darkness as it. So this is what simple corona. So that's why the name being kept for this virus as a corona virus. This is how they appear under electron microscope, the virus. The coating, which shows similar to that of a reminiscence of a corona, right? This is a different scanning microscope, electron microscope, which gives a picture of virons with a diameter of 200 nanometer average. So virons is maybe another word. It is a simple virus only, but a complete virus, an infectious virus can be called as a virons or virons. So this is a schematic diagram of a coronavirus with a particle of a spike protein, E protein, envelope protein, membrane protein, RNA genome, and so on, with a bilayer, all this. Probably all of you would have seen very boringly this picture. In all, lectures and probably TV news also, you might have seen. And particularly with the spike protein. So can you see that the genome and DNA is over here? So it is almost about the 25 to 35 kilobase per length. So this genome, with the overlapping species of an mRNA, it can give a varieties of transcripts for the different kinds of protein capsids with a different length. So they keep on producing these mRNA, which will be utilized by the host cell itself and then synthesize a particular protein, and then they get encapsulated and they synthesize the active virus and they start infecting the library cell as well. So I need to say like the overlapping gene as such. The coronavirus disease. So this causes some severe respiratory problems. Coronavirus causes severe respiratory problems such as corona respiratory syndrome, coronavirus pneumonia, coronavirus flu. All these were observed initially. We were not appreciated just because it is not so harmful as such. That's it. The disease caused by the members of a coronavirus family. It's not that a new virus has come. The coronavirus was there for a long time, but it was coming and going, coming and going. But we were not bothered much because there is a viral flu. We talk about. Viral flu comes and goes. Nothing to worry. The doctors always advise us to go. But half late, somewhere in 2002, there was an outbreak. The deaths were considerably more. Then we call it as what we call the SARS, severe acute respiratory syndrome. Based on that syndrome and the disease which is coming because of that virus and coronavirus, now it is alarm. So this was the first virus which we call it as a SARS-CoV virus, coronavirus. This was in 2002, somewhere in China, it happened, South Asia region. And later on, there is another same virus, a different one, Middle East respiratory syndrome, MERS. So this was abbreviated SARS, and this was abbreviated MERS, Middle East respiratory syndrome. This disease is caused because of a MERS COVID particular variant, so which was happened in 2012-14 outbreak, and since then it is recurring. And a coronavirus disease 2019, this made almost a chaos in the society. A disease caused by the SARS-CoV-2 first occurred in 2019, that's why we call it as a COVID-19. Otherwise, it is SARS coronavirus 2 is the kind of a variant. So this became what we call as a pandemic. All over the world it started spreading. So as you all are aware of this, the disease was first identified somewhere in Wuhan in 2019 in Hebei, China, and since then spread globally, resulting in 2019-18 pandemic. This is the city of Wuhan, which was covered in a BBC and all. And people started investigating too, and they have published even in the Nature too, by saying that a new coronavirus emerges from bats in China, devastates a young swine identified as the same region from a same bats as SARS coronavirus. SARS coronavirus and MERS coronavirus came. Now the SARS, another variant came up with, and this started killing a lot of swine initially, and then started infecting even the human as well. So this was the research being conducted and published too. How did this, the horseshoe bats, which are having initially, were able to come into these different organisms as such? The concept is that the spillover infection. So usually from one species to another species, the virus infection will not happen generally. So within the species, the viral infection which will happen. So here, what is happening is this virus is jumping from one species to another species, let's say. This is quite dangerous. So certainly such kind of things which happens in the nature, very rarely, this phenomenon we call it as spillover infection. So there are varieties of diseases like anthrax, Ebola, influenza, and Zika. All these were observed similar fashion, the spillover mechanism. Spillover events involve the virus overcoming several barriers to become a feasible in another species. So then slowly they get established. So just like this. Can you see over here? From birds to buffaloes, a deer or a livestock, and then moving to the human. So from species to species, there is a jump over of this virus that happens. Again, this cartoon also I have taken from the nature only. So this spillover mechanism, it happens not only with the COVID-19 alone, so many other viruses also had happened previously. So coming to this statistical figures and pictures of why we become so cautious about COVID-19 compared to any other kind of a virus. This has been shown over here. See, let's say all of them, this SARS virus or coronavirus, have come from the bats and bats to civet to man. So this is the SARS virus came in, the spillover mechanism. So can you see here, 8,000 people were affected, 800 were the fatality rate here. So the death rate is 10%. Then the MERS, 2012. Again, from a bat to camel to a man. Sorry, it is some in the Kannada version, which I converted for a long time. 800, around 2,500, 800 people were dying, let's say. That means almost 34%. COVID-19, bat to pangolin to a man. This is how the spillover has happened. So, whatever the data which I collected on that day, which was on 17th April 2020, right? This cartoon was prepared myself. So, nearly about, this was with a worldometer, okay, with the website here, you can see that the coronavirus happens in the pandemic, how much deaths are happening, how much infection is happening, all the data are available over here. You can just log on to this, even this is active, and you can still observe that, what is happening over there. Nearly about 6.7%. So looking at all these varieties of viruses, the most dangerous is the COVID-19. All of you agree?
No.
Correct? But here, by data, if you look at it, this appears to be more dangerous, right? MERS 34% death and 10% death and 6.7% death. So which one is the more dangerous? Certainly MERS, right? Now the tricky question comes.
Sir, but the data is for 2020. It's 23 now. So the data are comparing to 2020 and 23. Just COVID rate is high.
Very good analysis. Hypothesis is good. Need to be proved. So it's good. You made a very good hypothesis. The data which was shown in 2020, but if you look at now, this would be much more around than this. No, it is lesser than this. Now 3% something. Not even 3, even lesser than that now.
This thing is more contagious than all this.
Very good. That is the exact thing which happened. The COVID was the one which is more contagious. The spreading rate, probably you would have seen all YouTube and WhatsApp and social media, everywhere you would have seen wonderful stories being made and activities being made. So if you touch this, if you touch this, if you touch this, how this virus will get spreaded and how far it is this, all these stories being elaborated just to caution the people not to get infected. But unfortunately, it started spreading rampantly. Now, because of this, so certainly the death rate or overall death will be more if you take the overall the population. So somehow we need to curb it. So there was set up a program being conducted by World Health Organization, and then they started controlling to all different sources. So this is what exactly the CDC, Center for Disease Control, CDC was formed, and you have already, it was there for varieties of other diseases, particularly this disease was extensively dealt with, and how to prepare and protect yourself, what to do if you think you are sick, all this kind of captions, and to follow strict instructions, not to infect another person if you are infected. If you are not infected, how to keep yourself away from this infection. So all these things were extensively covered throughout the world with all kinds of India and so on. And later on, certainly, the treatment strategies, diagnosis were really particularly countries in India and even developing countries and undeveloped countries. Certainly, it is very, very difficult to control with a highly populated country, right? So the symptoms, CT scan and PCR. So these, PCR, the cost of PCR it goes up to 3,000 to 4,000 rupees. Affordable price is not there, right? So people will ignore it. So they will never go for diagnosis only. There are so many things and myths have started. So that is a totally different story today. People have kept even the goddess, corona goddess also. They started praying also, right? In some of the states. So all such things, right? And the treatments, at that time treatments were not there, and as I said, even the doctor started prescribing a simple, treatment for a common flu. So any flu was treated by using anti-inflammatory agents and drugs targeting to, particularly to the ACE receptor. People have started understanding the cause, how does it get infected through this receptor, angiotensin converting enzyme receptor. So to this receptor, okay, this spike protein goes and binds and then takes an entry. So now people started making, design the drugs and so on, which you will be hearing in the afternoon session, how to design a drug and develop it. That's it. RNA dependent polymerase, particularly this is an RNA virus. So certainly RNA dependent polymerase will make a copy of its DNA and then it will do all other kind of machinery molecules utilizing the host system itself, and it will package and then give complete active virus. And a chymotrypsin like the proteases and later on vaccines were developed. So like this, every stage, the medicines were started developing at every, pathway of this complete infection of the virus. Every stages were started developing. So little bit from the CDC itself again, the virus strain, as I understand it, students better need to understand. Usually in a common word, the people say like strain, virus strain, like this, different people, right? Viral strain. It's not my cargo, it is taken from the CDC. Okay, it is just like virus 2 happens like that. So viruses regularly create a new strains. So it's not a new thing. Any virus is bad. It regularly creates a new strain. New strains are created when virus make copies of themselves. When replicating, sometimes a mistake happens. So this is the kind of very natural phenomenon of the virus. Whenever it gets replicated, so usually there will be some kind of what we call as a mistake which happens. So those mistakes create a slightly different versions of the virus or a variant. So these are the viruses which we call it as a variant. For example, let's say MERS, SARS, or COVID, they are all variants. Even in the COVID-19, which we call it as a SARS-CoV-2, CoV-2, that also has got a variety of strains. So it's majorly that, just to clarify the terms that mutations, variants, and strains. Mutation is the process through which the SARS-CoV-2 virus challenges, particularly dealing with only the SARS which I'm telling you over here. The variants, which describes the version of the virus that are changed through a mutation from the original virus. And the strains being used in the same way as the word, the variant being used. Right? In a common language, we use all these terminologies, but they are same. But the thing is that the quality of, let's say, I should say, the inherent property of the virus, when it is getting replicated, there will be some mistakes. So, which brings about another strain when it is getting replicated. Right? So, as we all understand now, the phylogenetic background of the variants, right? So, varieties of, by then people started working out and trying to understand these viruses are present in a different kinds of phylogenetic tree or not. Yes, there are. In that way, you have come up with what is called alpha viruses, particularly which infect with these and civet and swine and human. And then beta viruses, which goes with bats to or other kind of a species, let's say, where again variants are there, and delta variants and a gamma variant, alpha, beta, and a gamma variant. These are the different kinds of SARS-CoV-2 we have come across. Right? So only when people started working with a complete genome analysis, they would have come to conclusion that this is a variant from this. Probably, I don't know whether you are good at bioinformatics or not. If you compare by using what you call the FASTA procedure, so if you are aware, so you can always compare the genome of another genome, or even a small piece of a DNA you can take and then compare the similarity search, right? And to get to know like how much similar they are, how much phylogenetically they are related to each other. So all such kind of exercises were done, and they have come up with RNA-dependent RNA polymerase. Till now, imagine now, we are started, my company started making a drug for RNA-dependent RNA polymerase. RNA-dependent RNA polymerase for a SARS-CoV particular variant. So by then you see that there is how many variations have happened now. My company will definitely go down. Got it? Because there are so many RNA-dependent RNA polymerase genes being mutated. So now if I design for this drug, so it doesn't work for another variant, which is having a different RNA-dependent RNA polymerase. So this is what happens exactly. So now you need to look at the quite stable, what you call it, the gene. For that you could decide, yes, it is possible. Otherwise, it's better to have other kind of what you call that mode of therapy, I should say. The mode of therapy is that we started looking at the spike protein and we started developing a vaccine for it. So if at all, if there is no vaccine at all, probably still there were many more deaths just because of the coronavirus. Right? So RNA dependent RNA polymerase. If you look at only this gene is this much of variants. If you look at the genes of other things, let's say even envelope protein, spike protein, or many other proteins. Sorry, genes. So there will be a lot many. See, another one is that the phylogenetic network of 160 SARS-CoV-2 genomes. The entire genome started analyzed by different parts of the world. And bad, particularly China, East Asia, USA, Canada, Europe, Australia, Mexico, and Brazil. See, so many variants. Within a span of 2 years, with a different, not even a span of 2 years, probably the moment when the SARS came into the picture or COVID-19 has started, every scientist started working on the complete genome project because the viral genome to complete is quite easy when compared to higher organisms like the human. Human genome project would take a long time, but the viral genome will not take much time. So they started working out and then they see that there is a greater variations which are happening. Right? So samples from different again parts and how variant they are. Sample from other countries, sample from China, sample from Wuhan alone and Shanghai being compared here. So again, this is one of the recent articles which I took it and then brought over here. So different variations are happening with the virus. It is a constant process. The mutational signature of the SARS-CoV-2. This is people have understood about the mutations and the rate of mutations which are increasing COVID-2 to COVID. So these are all technical details. I will not go in detail about it, but there is a variation which you can see over here, very clearly with those the variations which are happening, right? So beta virus, influenza, MERS, and SARS-CoV-2. So there is a one kind of observation being done is that the recombination blocks, which you will see over here. So there are some kind of what we call the recombination blocks on the ORS itself in the genome structure of a SARS-CoV-2. So this is something like mutation is the other hand, a single nucleotide getting varied, and the block itself will be taken into the another one. So the recombination process which happens. So all of you might be aware of recombination enzyme which does this, the changing the entire block and bringing it. Suppose this building has been taken and then kept it in the road. So it looks really awkward, right? A different structure altogether. If you look at that, the virus, it will be like that. So a different altogether, whenever it undergoes a recombination process, it gives a different kinds of strain. So it's quite difficult to design a drug for particularly SARS-CoV alone. So this is another one of the pictures you can see all over the world to how much variations have happened on this with the coronavirus alone. So the ongoing evolution is there. With all this ongoing evolution, the solution which we have found out is only through a vaccine right now. Does SARS-CoV-2 genome show any evidence that it is adapting to human hosts during the course of the pandemic? So this is another problem. Whenever the virus, which started, getting replicated, sometimes it gets integrated into our genome system itself. They will stay happily. So that may also happen. So that we will not have any kind of symptoms or something. Slowly it will get native. And then sometimes later, off late, it can revolt also. That is also there in a different kinds of viruses. Can be gained by the population genetics and analysis of COVID-2. So we need to go into the population genetics of this particular virus, whether it be. And people have started a global initiative sharing all influenza data, report back, and with the next strain database. So far, we had a next gen sequencing. Only the sequencing methodology has been changed in such a way that we do sequencing of a DNA and then make the what we call the faster sequencing. Now the next gen strain itself, the database. So we need to look at the varieties of viruses will be being different kind of strains and so on. So during that period, as I was talking about, we have made one mathematical model just to predict, the susceptibility, infection, recovery of the patients and all this. How quickly one can, get rid of this COVID-19. And it was very well established by many other people too, to say that, 2024 was the target earlier. With all parts of the gamma factors, those factors will be susceptible. So with the ongoing, so this graph also keeps on changing. So this virus may go off by 2024. By taking this virus with all other viruses earlier which came up. So that was a solution we tried. So that's all I should say with the coronavirus and the evolution of a small molecule to, let's say, a virus and how this virus is evolved. So thank you very much. So those, if you have any questions, I can try to answer.
Sir, the one doubt regarding that. We know that evolution of all organisms, it is a quite biological, natural phenomenon. So all the microorganisms present in this environment, they are also undergoing the process of evolution. So my doubt is that whether for the extinction of the human being, is it linked with the evolution of this, the most superbugs, the microorganisms? Suppose if they are go on undergoing the fast evolution because their generation means they are fast, they are having very short lifespan. When they undergo such a continuous and rapid mutations and evolution, in future, whether they are going to become the most enemies for the human life on this planet Earth.
Really wonderful question. And everyone has to think of this. So microbial world will going to exist. Our humans are going to survive. People, we are going to put in the same question. The evolution is such a way that the microbes are getting evolved, or even the viruses are getting so evolved rampantly. But humans are lost, right? But the thing is that when you go with a multicellular or a complex organism, the evolution goes in the time scale very, very short, and the small organisms can undergo much faster evolution. So that's why the viruses are getting more evolved and coming with the varieties of strains as such when compared to the other organisms, let's say humans. As rightly said, so we start concentrating only for earning only for food and shelter. We look at only the finance, not looking at exactly the business of science. So if we don't give a much interest to understanding about these viruses and microbes and all this world of evolution, I think the day will be apathetic. People will not even understand what is evolution at all. At least with this kind of a conference, probably you would have appreciated the microbes evolve and the virus evolve much faster and better than the human being. Right? If we survive only for our the financial aspects and giving the least importance for the actual science, the basic science of understanding of probably evolution and do work with that, the day will come, the microbes will knowingly or unknowingly they will take over. So certainly we need to give a much importance, at least the COVID blessing in disguise, all science people were appreciated after the COVID. So we have known like how these viruses come in and then kills all of us. Right? Irrespective of whether it's a top president or a prime minister or to a poor person, right? It doesn't leave anything. So that's we need to try to understand the pattern of our evolution and even the virus, microbes, all these things are very, very important to know. It's it's not that the microorganisms need to be killed or virus need to be killed. I'm not telling that. Some of the microbes are very beneficial too, but not everyone. So we need to know, like, at least pathogenic organisms, microorganisms, to, let's say, avoid. So we need to control that on our other means. And understanding the molecular pathway of infection and overcoming, killing the other organism. This is the fundamental property, I tell you, for any organism. Any organism in this world which try to survive itself first, killing the another. Not necessarily to killing the other, to survive is very important. If that other organism starts fighting, definitely the other will be killed. So this is the fundamental, principle of our life. So microorganisms or viruses, certainly they will overtake us one day if we are ignorant on those aspects. So at least if we know, like, how to get rid of this by making our own way, because the science has developed in such a way that, okay, we know, like, how to design a vaccine. If suppose they would have not known, like, how to design a vaccine properly, devastation would have been happened. I heard my grandfather died just because of a plague came. You know the plague, why did it come and all those things? It was published in 1992 in a science article. There is a one protein phosphatase called Yersinia phosphatase that goes and then rampantly dephosphorates all the signaling things which are happening in the cellular signaling. The kinases and phosphatases, they are the dynamics. They do the balancing signaling within the cell. If that is the kind of a cell, one is you can just Google it and then find it out. Yersinia protein tyrosine phosphatase. This was published in a scientific science journal, and this was discovered half late when the devastation has happened. So that is really utter negligence, or I'll say utter ignorance of the science. Probably today, within a 2 years of a span, we are able to design a vaccine for such a virus called COVID-19. Certainly, it is a wonderful achievement of a human being. Thank you. Thank you for wonderful question.
Everybody, please, if you have, don't think that it is a silly question, but the same question will be in the minds of others also. But this is a science, basic science. We have to try to get the concepts of the science. ಎಷ್ಟೇ ಸಿಲ್ಲಿ ಆಗಿದ್ರು ಕೂಡ ಪ್ರಶ್ನೆ ಕೇಳಲಿಕ್ಕೆ ಯಾರು ನಾಚಿಕೆ ಪಡ್ಕೋಬಾರ್ದು. ದಟ್ ಈಸ್ ದ ಬೇಸಿಕ್ ಕಾನ್ಸೆಪ್ಟ್ ಆಫ್ ಸೈನ್ಸ್. ನಾವು ಇವತ್ತು ನಾಚಿಕೆ ಪಡ್ಕೊಂಡ್ರೆ ಕೇಳೋದು ಹೆಂಗಪ್ಪ ಅಂತ ಮಾಡ್ಕೊಂಡ್ರೆ ನಾವು ಸೈನ್ಸ್ ಅಲ್ಲಿ ಮುಂದುವರಿಯೋಕ್ಕಾಗಲ್ಲ. ಪ್ಲೀಸ್ ಎನಿ ಸ್ಟೂಡೆಂಟ್ಸ್ ಯಾರಾದ್ರೂ ಕೂಡ ಕಾಮರ್ಸ್ ಇರಲಿ, ಆರ್ಟ್ಸ್ ಇರಲಿ, ನಿಮಗೆ ಇದ್ರ ಬಗ್ಗೆ ಏನಾದ್ರು ಪ್ರಶ್ನೆ ಇದ್ರೆ ಕೇಳಿದ್ರೆ, ಬಿಕಾಸ್ ಹಿಯರ್ ವಿ ಆರ್ ದ ಎಕ್ಸ್ಪರ್ಟ್ಸ್ and they will give the concrete proof answer for that. Thank you. Thank you, sir.
Thank you very much.
Thank you so much, sir, for giving us brief information. Now I would like to call our principal, sir, Dr. Arunkumar sir, to give presidential remarks.
Respected Chief Guest of this National Conference, Professor Devraju, yes. I'm very much happy and proud because Devraju sir also belongs to the same university where I learned and where I graduated and post-graduated and got the PhD also. That is my alma mater, Karnataka University, Harvard. Karnataka University, Harvard has produced a number of eminent academicians, researchers, and they have become world famous also. Karnataka University has actually was founded by Wrangler Pauke, who has made this Karnataka University globally a recognized university. And every year it is producing number of doctors, doctorates, and number of postgraduate and graduates. Now they are contributing to the basic science or any other branch of the education. Where we hails from that department, biochemistry. Actually, the biochemistry in the Karnataka University started from the botany department, and whereas later it has been converted into biotechnology. And I am the first batch student to get the biotechnology from the Department of Life Science. Then the biochemistry department was established, and it has become so popular that number of patents have been applied and they have been awarded patents by great eminent scientists who are there in the Karnataka University, especially Swami sir and my research guide and my mentors, Dr. Shivkumar Swami from who has got his PhD from UK, and all they have contributed to the Karnataka University. So still the Karnataka University is very popular in the academic growth. So such a personality, Dr. Devaraju, is also from the Karnataka University where he is working. Really it is a very nice moment to share with us some of the opinions with him. And now this is a topic which is very, very common and a basic, the title has been given by the mentor of our UGC style component. That is V. Prakash sir. He is the founder, he is the former director of CFTRI. He is the mentor of this UGC style component. He is the Padma Bhushan also, and he is the advisor to the Prime Minister Narendra Modi for Science and Technology. Such a great and great personality is the mentor for our college, and they are getting the suggestions from him. And Dr. Devan sir, we have given you the only the superficial touch to the evolution and evolutionary infectious diseases. But here I would like to share that he's only explained about the outlines, but if you go and more deeper, it will become completely a molecular biology. But I am standing before you to do the presidential mark, so I have to narrate about what is this evolution and what are the infections, how the correlation is there. ನಾವೆಲ್ಲರೂ ತಿಳ್ಕೊಂಡಾಗ ಈ ಭೂಮಿಗೆ ನಮ್ಗೆ ಎಷ್ಟು ಹಕ್ಕಿದೆನೋ ಅಷ್ಟೇ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ಸ್ಗೂ ಕೂಡ ಅಷ್ಟೇ ಹಕ್ಕಿದೆ. Every creature of this planet Earth, they have the same right to exist here, first to origin here, to exist here, to survive here, and every organism is fighting every day in order to get the food, the shelter, and reproduction. ಎಲ್ಲರೂ ಕೂಡ ಬರ್ದಾಗ್ತಾ ಇರೋದು ಬೇಸಿಕಲಿ ನಮ್ಗೆ ಗೊತ್ತಿಲ್ಲದೆ ಬರ್ದಾಗ್ತಾ ಇರುವಂತ ಸಂಗತಿ ಏನಪ್ಪಾ ಅಂದ್ರೆ ಮೂರು ವಿಷಯ. ಅದು ಒಂದು ಫುಡ್, shelter, and reproduction. The last one is reproduction. Unknowingly we are reproducing. ಯಾರು ಹೇಳ್ಕೊಡ್ಬೇಕಾಗಿಲ್ಲ. The population of every creature is actually it is growing in the geometrical ratio, not in the arithmetic ratio. Try to understand the natural resources which are available, they are in the arithmetic ratio. Whereas the population of the every creature is increasing in the geometrical ratio. Andre into multiply ಆಗ್ತಾ ಹೋಗುತ್ತೆ. So where there is we have now we actually understood plague ಅಂತ ಬಂತು. When the plague is arose, a number of the people they have lost their lives because the science and technology it was not in a very the sufficient or the potential way at that time. The reason is that our plague barbecare only one muಖ್ಯ how the nature is doing all this. Only thing is that always nature is trying to balance the entire this ecosystem. ಅರ್ಥ ಮಾಡ್ಕೋಬೇಕು. ಈ ಭೂಮಿಗೆ ಒಂದು ಶಕ್ತಿ ಇರೋದೇನಪ್ಪಾ ಅಂದ್ರೆ ಯಾವಾಗಲೂ ತನ್ನಷ್ಟಕ್ಕೆ ತಾನೇ ಬ್ಯಾಲೆನ್ಸ್ ಮಾಡ್ತಾ ಇರುತ್ತೆ. ಯಾಕೆ ಈ ಡಿಸೀಸ್ ಬರಬೇಕು, ಡಿಸೀಸ್ ಯಾಕೆ ಬರಬೇಕು, ಬರಬಾರ್ದು ಯಾಕೆ ಅಂತ ವಿಚಾರ ಮಾಡಿದಾಗ ಇಟ್ ಇಸ್ ಅ ಕ್ವೈಟ್ ನ್ಯಾಚುರಲ್ ಫಿನಾಮಿನಾ. ಭೂಮಿಯಲ್ಲಿ ಆಗ್ತಕ್ಕಂಥ ಈ ಸಂಗತಿಗಳು ಏನಪ್ಪಾ ಅಂದ್ರೆ ಸೊ ದೇರ್ ಇಸ್ ಟು ಬ್ಯಾಲೆನ್ಸ್ ದಿಸ್ ದ ಥಿಂಗ್ಸ್ ಆರ್ ಹ್ಯಾಪನಿಂಗ್ ಹಿಯರ್. ವೇರ್ ಇಟ್ ಕಮ್ಸ್ ಟು ದಟ್ ಎವಲ್ಯೂಷನ್ ಹೇಳ್ತಾ ಇರಬೇಕಾದ್ರೆ ಸರ್ ಯು ಹ್ಯಾವ್ ಸೇಡ್ ದಟ್ actually when the earth was created it was in a hot gaseous form. ಒಂಥರ gaseous ಬೆಂಕಿ ಅಂತ ಒಂದೇ ಆಗ್ತಾ ಇತ್ತು, and it has gone cooling itself and it took many millions years. When it has started to cool down, more than 40 years there was a torrential rains, ಆ ಭೂಮಿ ತಂಪಾಗಬೇಕು, ಆ ಸೂರ್ಯನಿಂದ ಬಂದಂತ ಡಿಪೆಂಡ್ ಆದಾಗ ಆ ಬೆಂಕಿ ಉಂಡೆ ಏನಾಯ್ತು ಅದು ತನ್ನಷ್ಟಕ್ಕೆ ತಾನೇ ತಂಪಾಗಬೇಕಾದ್ರೆ that all the gaseous molecules which were there, they started to condense themselves. Then the cooling process has started and it has converted into the torrential rain. What was mಳೆ ಆಗ್ತಾ ಇರಬೇಕಾದ್ರೆ it is hypothesis. There is no evidence for that. So hypothesis says that when the torrential rains are happened, that water used to, that gaseous molecules used to condense into the water, and as much as they used to reach the surface of the earth because of the high temperature, they used to once again evaporate. And finally they used to make the cloud and the rain. So this kind of the cycle of torrential rains that has finally made this culture filled with the water, where all these elements, what they call them, more than 180, they were all dissolved in that liquid that is called as the primordial soup. So it was like a hot dilute soup it was there. And there was a chance of resource of the energy. ಯಾವುದೇ ಒಂದು ಕ್ರಿಯೆ ಆಗಬೇಕು ಅಂದ್ರೆ ಅಲ್ಲಿ ಶಕ್ತಿ ಬೇಕಾಗುತ್ತೆ. There was a tremendous source of energy from the sunlight as well as from the background radiations were there. ಆ ಭೂಮಿಯಲ್ಲಿ ಇರ್ತಕ್ಕಂಥ ಬಹಳಷ್ಟು ರೇಡಿಯೇಷನ್ ಮಾಲಿಕ್ಯೂಲ್ಗಳು ವಿಕಿರಣ ಬಿಡ್ತಾ ಇರಬೇಕಾದ್ರೆ that all the radiations were available in the background of the earth as well as from the sun because there was no ozone at all. So then this magic thing is happened, that it is called as magic broth and happened. Primordial, the soup has converted into the magic broth and all the chemicals they started to attach with each other. Now molecule attach of energy ಸಿಕ್ಕಾಗ ಒಂದು bond ಆಗಿ they started to convert to the macromolecules and then they became into the primordial earth where some of the very ancestral organisms are created. ಮೊದಲು ಹುಟ್ಟಬೇಕಾಗಿದ್ದು ಏಕಕೋಶ ಜೀವಿ, ಅದು single celled organism which were there. They are also called as the prokaryotes. Probably ಎಲ್ಲರೂ ಓದಿರ್ತಾರೆ PUC prokaryotic organisms. There was nucleus was not there, and then they slowly converted into eukaryotes. Then the things are become complex. So ಅರ್ಥ ಮಾಡ್ಕೋಬೇಕಾದ್ರೆ always there is the evolution process is going on. Till today the evolution process is going on. So that single celled organism then undergoes the process of the evolution, and today we have become the most very strong human being that is a complex elements. We call them as ಎರಡು ಕಡೆ ಹೋಗ್ತಾ ಇರ್ಬೇಕಾದ್ರೆ ಈ ಕಡೆ ಪ್ಲಾಂಟ್ಸ್ ಬರ್ತಾ ಇತ್ತು, ಈ ಕಡೆ ಎನಿಮಲ್ಸ್ ಹೋಗ್ತಾ ಇತ್ತು, ಎರಡು ಕೂಡ ಒಂದಕ್ಕೊಂದು ಅನುರೂಪವಾಗಿ ಬೆಳೀತಾ ಇರಬೇಕಾದ್ರೆ there are different stages ಆಗ್ತದೆ. And then ಇವೆರಡೂ ಬಿಟ್ಟಾಗ ಪ್ಲಾಂಟ್ ಕಿಂಗ್ಡಮ್ ಅನಿಮಲ್ ಕಿಂಗ್ಡಮ್ ಬಿಟ್ಟಾಗ ಇನ್ನೊಂದು ಕಿಂಗ್ಡಮ್ ಇದ್ದು that is called as the microbial world. And all the microbial ಅವೇ ಮೊದಲು ಹುಟ್ಟಿದಂತ ಪ್ರಾಣಿ creatures, these microorganisms have been created first, but still they are in the microbial condition. ಯಾರು ಎಲ್ಲಿ ಗ್ರೋತ್ ಆಗುತ್ತೆ ಅದೆಲ್ಲ ಗ್ರೋತ್ ಆದ್ಮೇಲೆ still the microorganisms are there in this planet Earth everywhere. They are called as cosmopolitan in habitat. Cosmopolitan is that they are present in the air, water, even in the soil, even in the hot water springs, and even in the top of the Antarctica also microorganisms are there. And even to surprise, even the microorganisms are present in our body itself. ನಮ್ಮ ಬಾಡಿಯಲ್ಲೂ ಕೂಡ ಇದೆ. There we can call them as the microflora of the buccal cavity or the microflora of the stomach or the microflora of the intestine. So ಎಷ್ಟೊಂದು ಒಳ್ಳೆ ಜೀವಿಗಳು ಇದೆ ಅಂತ ಹೇಳಿದ್ರು ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಅವೆಲ್ಲ ಕೂಡ ನಮ್ಮ ದೇಹದಲ್ಲೂ ಇದೆ, ಹೊರಗಡೆ ಇದೆ. But there is always a fight between the microbes and all these plants and animals. ಎರಡು ಕಡೆ ಪಾರ್ಟಿ ಇದೆ, ಈ ಕಡೆ ಒಂದು ಪ್ಲಾಂಟ್ ಕಿಂಗ್ಡಮ್, ಎನಿಮಲ್ ಕಿಂಗ್ಡಮ್, ಈ ಕಡೆ ಮೈಕ್ರೋಬ್ಸ್ ಯಾವಾಗಲೂ ಕೂಡ ವಾರ್ ಆಗ್ತಾ ಇರುತ್ತೆ. Those that become strong and they will just overcome that, they will bypass them, they will become strong. But ಯಾಕೆ ನಾವು ಬದುಕ್ತಾ ಇದ್ದೀವಿ ಅವ್ರು ಹೇಳಿದ್ರು, ಒಂದು ಒಳ್ಳೆ ಕಾನ್ಸೆಪ್ಟ್ ಹೇಳಿದ್ರು, and we have solved it within 2 years. Within 2 years we have solved that COVID, the SARS pandemic situation. It's because the human being is called as the master man or the master human being. We have a brain which is not available in any other the simple living organisms. ಗಿಡಗಳಲ್ಲಿ ಬೆಳೆಯಲಿಲ್ಲ, ಕೆಲವೊಂದು ಎನಿಮಲ್ ಬ್ರೈನ್ ಇದ್ರೂ ಕೂಡ ಅಷ್ಟೊಂದು ವ್ಯಾಪಕವಾಗಿ ಎವಲ್ಯೂಷನ್ ಆಗಿಲ್ಲ. And this is only because of the brain capacity we are able to rule this planet Earth. But ನಾವು ಹೇಳಿದ ಹಾಗೆ ನಾವು ಇದನ್ನ ಓವರ್ಕಮ್ ಮಾಡಿ, if we are not degrading this nature and natural resources, and in the time to come or the days to come where these microbes once again they become dominant over the human being. One example ಕೊಡ್ತೀನಿ, ಇಲ್ಲಿಗೆ ಬರಬೇಕಾದ್ರೆ ಕಾರಣ ಏನಪ್ಪ ಅಂದ್ರೆ ಬಾಂಬು ಪ್ಲಾಂಟೇಶನ್. ಅವಾಗ ಬಾಂಬು ಒಂದು ವಿಶೇಷ ಏನಪ್ಪ ಅಂದ್ರೆ ಈ ಬಾಂಬು ಅಂದ್ರೆ ಬಿದಿರು, ಅದು 30 ವರ್ಷಕ್ಕೊಮ್ಮೆ ಫ್ಲವರಿಂಗ್ ಆಗುತ್ತೆ. ಈ ಗಿಡ ಸುತ್ತಬಾರ್ದು 30 ವರ್ಷಕ್ಕೊಮ್ಮೆ ಫ್ಲವರ್ ಆಗ್ತಾ ಇರ್ಬೇಕಾದ್ರೆ ಫ್ಲವರ್ ಆಗಿ ಸೆಟ್ ಆಗಿ ಸೀಡ್ಸ್ ಆಗುತ್ತೆ. ಆ 30 ವರ್ಷಕ್ಕೊಮ್ಮೆ ಬಿಟ್ಟರೆ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಮೀನ್ಸ್ it stops its asexual reproduction and it starts sexual reproduction. ಆ ಸೆಕ್ಷುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಆದಾಗ ಫ್ಲವರ್ ಆಗುತ್ತೆ, ಫ್ರೂಟ್ ಆಗುತ್ತೆ, ಸೀಡ್ಸ್ ಆಗುತ್ತೆ. ಅವಾಗ ಇಡೀ ಈ ಭರತಖಂಡದಲ್ಲಿದ್ದಂತ ಎಲ್ಲ ಬಾಂಬುಗಳು ಎಟ್ ಎ ಟೈಮ್ ಏನಾಗುತ್ತೆ ಫ್ಲಾರಿಂಗ್ ಆಗುತ್ತೆ, ಅವೆಲ್ಲ ಸೀಡ್ಸ್ ಆಗುತ್ತೆ, ಅದಕ್ಕೆ ಬಿದಿರು ಅಕ್ಕಿ ಅಂತ ಹೇಳ್ತೀವಿ. ಆ ಬಿದಿರು ಅಕ್ಕಿ ಕೆಳಗಡೆ ಬಿಟ್ಟಾಗ ಅದನ್ನ ತಿನ್ಲಿಕ್ಕೆ ದ ಪಾಪ್ಯುಲೇಷನ್ ಆಫ್ ದ ರಾಟ್ಸ್ ಅಂಡ್ ಮೈಸ್ ಹ್ಯಾಸ್ ಬೀನ್ ಇನ್ಕ್ರೀಸ್ಡ್. ಅದು ತಿನ್ಲಿಕ್ಕೆ ಏನಾಯ್ತವಾಗ ಆ ಇಲಿಗಳ ಪಾಪ್ಯುಲೇಷನ್ ಎಷ್ಟಪ್ಪ ಹೆಚ್ಚಾಯ್ತು ಅಂದ್ರೆ ಎವ್ರಿವೇರ್ ದೇ ಬಿಕಮ್ ಡಾಮಿನೆಂಟ್. ಸೊ ದೆನ್ ದೇ ಹ್ಯಾವ್ ವಾಟ್ ದಿ ಒನ್ ಆಫ್ ದ ವೈರಸ್ ಅಂಡ್ ದಟ್ ಹ್ಯಾಸ್ ಕ್ರಿಯೇಟೆಡ್ ದ ಹವಾಕ್ ಆನ್ ದ ಪ್ಲಾನೆಟ್. ಅವಾಗ ಪ್ಲೇಗ್ ಅನ್ನುವಂತ ರೋಗ ಬಂತು. ಅವಾಗ ಇಷ್ಟೊಂದು ಸೈನ್ಸ್ ಡೆವಲಪ್ ಆಗಿರಲಿಲ್ಲ. ದಟ್ ಮೋಸ್ಟ್ ಆಫ್ ಅವರ್ ancestors they have lost their lives. So it is what we can say. It is always the tug of war is there between the plants, animals, and the microorganisms. But it is only because of the basic science what we are doing continuously the research. Only because of that reason we are able to resist any kind of the microorganism and we are able to dominate this planet. ಆದರೆ ಮುಂದೆ ಬರ್ತಕ್ಕಂಥ ಜನರೇಷನ್ ನಾವು ವಿಚಾರ ಮಾಡಬೇಕು. If you are using that all the natural resources in a very harmonious condition, if you are using it in a very good condition, we can survive, we can hand over the next generation. ಇಲ್ಲ ಅಂದ್ರೆ in future probably the worst things are to be happen to the human being. So that's why please my humble request is that all the common man we should understand the biological concept. The biological concept ಅರ್ಥ ಮಾಡ್ಕೊಂಡ್ರೆ ನಾವು ಇಷ್ಟೊಂದು ಚೆನ್ನಾಗಿ ಕೋವಿಡ್ ಅನ್ನ ಫೈಟ್ ಮಾಡಿ ಎದುರಿಸಲಿಕ್ಕೆ ಕಾರಣ ಏನಪ್ಪಾ ಅಂದ್ರೆ ಎವ್ರಿಬಡಿ ಹ್ಯಾಸ್ ಬೀನ್ ಎಜುಕೇಟೆಡ್ ಇನ್ ದಿಸ್ ಕೋವಿಡ್ ವೈರಸ್. ಯಾರು ಕೇಳಿದ್ರು ಕೋವಿಡ್ ವೈರಸ್ ಅಂದ್ರೆ ಚೆನ್ನಾಗಿ ಹೇಳೋದು ಭಾಷಣ ಮಾಡ್ತಾರೆ. ಯಾಕಪ್ಪ ಅಂದ್ರೆ ಬಿಕಾಸ್ ದೇ ಆರ್ ಟೋಟಲಿ ಎಜುಕೇಟೆಡ್. So in the same way, if you are educated in all the basic science subjects, especially basic biological subjects, definitely we can survive in a very long way in a healthier way. This is my only remarks I should say, and this is the national conference. The message goes to all the youngsters is that we have to please have the education in the basic science, especially in the America and the developed countries like Europe. There we know that every human being they have to study the biology because only it helps to us how to grow and how to allow the other organisms to survive on this planet. So that's why this is only my request to all the participants over here. Please respect our environment, respect all the organisms. ಎಲ್ಲ ಪ್ರಾಣಿಗಳಿಗೂ ಸಸ್ಯಗಳು ಎಷ್ಟೊಂದು ಹಕ್ಕಿದೆನೋ ನಮಗೆ ಎಷ್ಟು ಹಕ್ಕಿದೆನೋ ಅಷ್ಟೇ ಹಕ್ಕು ಅವು ಕೂಡ ಇದೆ. ಅವೆಲ್ಲರೂ ಕೂಡ ನಾವು ಬದುಕಲಿಕ್ಕೆ ಅವಕಾಶ ಮಾಡಿಕೊಟ್ರೆ ಅವಾಗ ಎಕೋಸಿಸ್ಟಮ್ ಅಂದ್ರೆ ಎನ್ವಿರಾನ್ಮೆಂಟ್ ಚೆನ್ನಾಗಿ ಇರಲಿಕ್ಕೆ ಸಾಧ್ಯ ಇದೆ. Then we can survive for a longer period or even we can hand over to the our next generation. With this once again I'm very happy to see that Dr. Devaraj sir has enlightened us how this molecular biology is actually helping us to fight against any kind of superbugs. One of my senior professor and our mentors to the KLE Society, Dr. Kopate sir, is a very good pharmacologist. Because I also work in the most of the pharmacology. My most of the research papers, more than 10 papers in the pharmacology, where that Professor Kopate used to say that the superbugs are there, they are going to rule this planet. Only thing is that we have to aware and we have to plan and we should be ready in order to fight against the superbug. ಇವತ್ತು ಕೋವಿಡ್ ಇಲ್ಲ ಎಂಡ್ ಆಗಿಲ್ಲ. ನೆಕ್ಸ್ಟ್ ಇನ್ನೂ 1 ವರ್ಷ 2 ವರ್ಷ 10 ವರ್ಷ 50 ವರ್ಷಕ್ಕೂ ಮತ್ತೊಂದು ಬೇರೆ ರೀತಿಯ ವೈರಸ್ ಬರ್ಬೋದು. ಬಟ್ ನಾವೆಲ್ಲರೂ ಕೂಡ ಜಾಗೃತಿಯಾಗಿರ್ಬೇಕು ಅನ್ನುವಂಥ ಮಾತನ್ನ ಹೇಳ್ತಾ once again I am very much thankful to all the participants and all the resource persons and all the online participants who are thank you very much.
Thank you so much, sir, for giving us great information. Now I would like to call Narayan sir to give word of thanks.
Thank you very much. Good afternoon to one and all. Thank you to such a prayer that cannot see effect. Here, today, we are assembled and discussed about these national seminar, evolutionary biology and the diseases. First of all, I would like to thank the first session, the speaker, delegate, Dr. K. Devaraju, very beautifully explained about this evolutionary biology and diseases. Thank you very much. My sincere thanks to our beloved principal, Dr. Arun Kumar B. Sonapura sir, for the continuous support and constant support and leadership. Thank you. I would like to thank to our head of the department, Dr. Tejaswini like to thanks to our IPIC coordinator, Chandrasekhar Prasad. Thank you very much. Thank you. Thank you very much.
Now there is a tea break from 11:30 to 11:45. I request all of you back by 11:30. Tea time for 10 minutes and one more.
ಪೋಸ್ಟರ್ ಬೇರೆ ಕಾಣಿಸ್ತದೆ ಇಲ್ಲ.